Good morning, my dear students. Today we are discussing about the new chapter, so that is blastulation. After the process of cleavage and the next embryonic development, what we find that what we call it as blastulation. So, first, embryonic development. We have already discussed this, that is the cleavage, which is the first major embryonic development, which we call it as the rapid succession of cell division. So, by cleavage, it creates a multicellular embryo from the zygote. Here you cannot find any growth, whereas you can see the 2 cell into 4 cell, 4 cell into 8 cell, and then 16 cell to the next stage, what we call it as the morula stage. So, morula stage, an early stage in the post-fertilization development when cells have rapidly mitotically divided to produce a solid mass of cell. So, it may consisting of 16 or more contained within the zona pellucida. The mulberry appearance, that what we call it as the word, that is morula stage. The morula stage, it is the final stage prior to the formation of a fluid filled cavity called the blastocele cavity. The cells of the morula are segregated into the inner mass cell and the outer cell mass, and it consisting of a hollow to form a, that is the blastocyst. So here you can see the blastula and the blastocyst, where that means the morula give rise to blastula and the blastocyst. Blastocyst, it is the mammalian blastula. So it will start or the precursor from the, that is the zygote. So next, very important, after the cleavage, so the cleavage, then the next process, what we call it as the morula. Next, it is the blastula. The development of multicellular organism begins from a single-celled zygote which undergo rapid cell division to form the blastula. The rapid multiple rounds of cell division are termed as cleavage, and after the cleavage there are about 100 cells, and the embryo and that stage, what we call it as the blastula. See the blastula in this picture, you can see that it is a spherical layer of cells, that means the outermost, which is surrounded by a fluid filled or yolk filled cavity, that what we call it as the blastocele. So mammals at this stage, they form a structure called the blastocyst, and which is characterized by the inner mass, and that will be distinct from the surrounding blastula. So during the cleavage, what happens, the cell they starts to divide without increase in mass. That means there is no growth at all. That means one large single celled zygote, they get divided into multiple smaller cells. So each cell within the blastula, we call it as the blastomere. So in the blastula of an animal, you can see the blastoderm, outermost layer, with a cavity, that is blastocele, and the cell, what we call it as blastomere. So the process of formation of the blastula, we call it as the blastulation. So this blastula will be different in case of different animals, though that will depends on the size, along with that the amount and the distribution of the yolk. You know how exactly the yolk has been distributed in different animals. So on that basis, so there are so many different types of blastula can be recognized in different animals, like coeloblastula. You can see in the echinoderms, amphiaxis and amphibians. Steroblastula, that is insects, some nereis, worms like nereis, mollusks, and also in certain fishes. Discoblastula in the fishes, reptiles, and also in birds. Blastocyst, it is the blastula stage of mammals. Periblastula means the most arthropods which have a centrolecital egg. That means example, very good example for the centrolecital egg, those are the insects. So these are the different types of blastula you can find in different animals. So next, that is the next what you have in your syllabus, that is you should make the comparative study of the blastulation what you found in the frog as well as in the chick. So before going to the comparative study, first we'll start with the cleavage in the frog. First, the process embryonic development starts with the cleavage, then it moves with the morula, and then it moves with the, that is, blastula. So the first cleavage of frog's eggs was observed by Swammerdam in 1738. The entire process of cleavage in frog's eggs was studied by Prevost and Dumas in 1824. In frog's eggs, the cleavage is holoblastic and unequal. Holoblastic means when an egg divides completely, we call it as the holoblastic, and in a holoblastic, that is the holoblastic equal and holoblastic unequal. That means the cells after the division, what you find, there are different types. That means the size of the cells that will be different. Some will be small and some will be bigger one. So that what we call it as unequal. So when you move on with the cleavage, the first cleavage, just look into the picture. The first cleavage plane is meridional and the second cleavage furrow is again meridional. That means it will bisect both the pole. In the next cleavage, it is horizontal, which is above the equator, resulting in micromeres and macromeres. That means two types of blastomeres or the cells they are found. The smaller ones what we call it as the micromeres, and the larger one what we find it is the macromeres. The fourth set of cleavage planes are meridional, but they are double in nature, they are unequal. The fifth cleavage is horizontal, or we can call it as a latitudinal, and it is also double, dividing the micromeres as well as the macromeres, so that the four tiers of blastomeres they are formed. So as a result of further cleavage, you know that next stage what we find, a ball of several small blastomeres, that what we call it as the morula stage. So next, when you come for the blastulation in frog, at the end of the cleavage, a solid balls of cell give rise to blastula, which consisting of number of blastomeres. The characteristic feature of the blastula stage are the presence of a well-defined cavity, that what we call it as the blastocele, which is a characteristic feature of the blastula. This is the beginning of primary body cavity. The process of formation of blastula is known as blastulation. The blastula of the frog is also known as the amphiblastula, as the cavity is confined to the animal pole. In this picture, you can make out the two important pole, animal pole and the vegetal pole, where the blastocele which is confined to the animal pole. The vegetal pole, it is composed of solid mass of non-pigmented yolky cells. In the 32 cell stage, the blastula consists of a single layer of cells and is called as early blastula. The pigmented cells, that is micromeres, they are found in the anterior half, while the yolky megameres, that means the larger cells, are present in the posterior half. Moving on, the blastocele lies entirely in the anterior region. The blastula of the frog is a halo, and they have a well developed blastocele, and that type we call it as the coeloblastula. The type of blastula what you find in the frog, it is the coeloblastula. So as the segmentation proceeds, the number of cells in the blastula increases, so also the blastoceles also increases its size. The floor of the blastocele is flat while the top portion is arched. You can look at this picture. The roof, it is made up of 3 to 4 layers of pigmented micromeres, small cells, while the floor it is formed by yolk megameres. Between the micromeres and the megameres and along the equator is found a group of cells which are intermediate in size. That means between megameres and micromeres. These cells constitute the germ ring. The germ ring is formed in the region of Greek crescent. So that is where you can see in the frog's egg, when we are discussing about different types of egg, we have discussed about the region what you find in the amphibian egg. So this is about the cleavage and the blastulation what you find in the frog. So when you come to the cleavage in the chick, so here the cleavage it is restricted to the blastodisc and the yolk remains uncleaved. Such cleavage is called meroblastic or discoidal cleavage. That means it has not divided the entire egg, so it is called as meroblastic. The central part of the egg, that what we call it as the blastodisc, when you look at the chick egg, you can clearly notice where exactly the blastodisc will come. The central part of the blastodisc is whitish and circular. So it is surrounded by darker marginal zone which is known as periblast. So they get merged with the white yolk. So that is the where you can see in the yellow yolk and the white yolk you can see in the hen's egg. So when it undergoes for cleavage, first cleavage after 5 hours of fertilization, the first cleavage will appear. It is confined to the center of the blastodisc. It is meridional. It cannot completely divide the blastodisc. Here the blastomeres they are not formed. When you go for the second cleavage, it takes place at right angles to the first. Even because of the second cleavage, clear blastomeres they are not formed in case of chick. Third cleavage, it is vertical and parallel to the first division. It is in the two sides of the first division. As a result of this division, 8 blastomeres are formed, but they do not show the boundaries. So fourth cleavage, it takes place in such a way that 8 central blastomeres and 8 peripheral blastomeres, that means which are at the margin. So they are also called as marginal blastomeres will form. Only at this stage of division definite cells they are formed. Till then there is no, the blastomeres they are not at all formed. The central 8 cells they completely separated from the yolk. So you can see 8 peripheral blastomeres and also 8 central blastomeres at the end of the fourth cleavage. In these cleavage, the furrows do not extend right up to the edge of the disc, so that the blastomeres which are present in the central area have distinct boundaries, and those have the outer area have no outer boundaries, but they get merged into the unsegmented syncytial periblast. So the marginal syncytial periblast brings the yolk and growing mass of the cell in the blastoderm into the nutritive contact. So here what happens, if you look at this picture, the central cellular area they start to expand by the addition of the cells which are present at the margin or the periphery. Later, the horizontal division takes place and the central area becomes 2 or 3 cell thick. So here, this gets separated by the underlying yolk by a space what we call it as subgerminal cavity, or you can call it as the blastocele. See here in this picture you can clearly make out the blastocele or the subgerminal cavity. So later on which develops either by splitting or separation of upper layer from the lower layer that retains the connection with the yolk mask. So here in the blastulation of the chick, the cells will undergo further division quickly. Hence, the segmentation cavity, consisting of mass of cells, will be present in several layers. These cells, they have a complete boundaries. The cells present towards the periphery are not separated from the yolk. They are called as marginal cells. Here in this picture, you can clearly observe the marginal cells. This region we call it as zone of junction. So here there are two areas they are formed, that is area pellucida and area opaca. The central cell mass of blastoderm will be 4 or 5 layers. They are lifted from the yolk. Hence, the central part of the blastoderm is free from the yolk and this region is transparent. So, that what we call it as the area pellucida, which is destined to become the embryo proper. At the zone of junction, the cell come in contact with the region what we call it as opaca. That region is called area opaca, which gives to extra embryonic structure. So here in this picture you can clearly make out the central subgerminal cavity, or you can call it as the blastocele, and the two important cells what we have said about the central cells and the marginal cells, and there is a yolk which has been lifted and the two areas have been formed, that is area pellucida and area opaca. So this area opaca, they later becomes differentiated into 3 more or less distinct zones. The blastomere produced from the original periblast constitute a germ layer or a germ wall. Due to the addition of more and more blastomeres to the peripheral region, the outer ring called the margin of overgrowth is formed. And the blastomeres which are formed in this region, they have no well-definite boundaries. So, inner to the germ wall area and lying in close contact with the yolk or a group of cells which without a complete cell boundaries. So, this region what we call it as the zone junction. The blastoderm at this stage contains 2 types of cells, that is relatively large yolk-laden blastomeres and small yolk-free blastomeres. Now follows a segregation wherein the yolk-rich blastomeres, they get gradually accumulated at the undersurface of the blastoderm, leaving the smaller yolk poor blastomeres at the surface. So this upper layer is called the epiblast and the lower layer we call it as the hypoblast. So here you can see in the center you can see the blastocele, and the upper part we call it as the epiblast and the lower layer we call it as the hypoblast. So in this picture you can clearly see a narrow cleft called the blastocele appears between the epiblast and the hypoblast. So this separation of epiblast from the hypoblast, we call it as the delamination. The cleavage results in the conversion of a blastodisc into a disc-shaped blastula called discoblastula, so which floats at the top the yolk mass. So here this is what the blastulation and the cleavage what you found in the chick. So the type of blastula what you found, it is the coeloblastula, whereas in the chick it is the discoblastula. So now your job is to what is exactly the question here in this chapter, that is you have to bring out the differences between the blastulation of frog and also the chick. Thank you.